Today I will be discussing and analysing the following topics. What is autism? The phrase autism spectrum disorder includes autism, Asperger's syndrome, and processive developmental disorder. Autism spectrum disorder is a disorder that impacts an individual's capability to interact with the world around them. This disorder is comprised of extensive varying unique levels and ranges of severity. It is a complicated disorder. Individuals on the autism spectrum frequently encounter difficulties with social communication and collaboration and have restricted repetitive patterns of behaviors, interests, or activities. In our world today, autism impacts roughly 1 in 100 to 1 in 110 young children. Statistics show that males are 3 times more likely to be impacted than females. There is no cure for autism, however, there are many treatments that aid this disorder. I will be bringing to life the transcranial direct current stimulation during this presentation. Unfortunately, autism does not have a certain combining mechanism at either the molecular cellular systems level. The physiological mechanisms are yet to be entirely realized due to its intrinsic nature. Nevertheless, there are a number of hypotheses to determine the psychological mechanisms of autism. The mirror neuron hypothesis of autism suggests that social and communication challenges are caused by the differences in mirror neuron activity. Mirror neurons were discovered in 1996 by Giacomo Risolti. The mirror neuron system plays a key role in our ability to empathize and socialize with others and is comprised of various means of understanding speech, understanding the meaning of other people's actions and minds. Recent empirical research determines that mirror neuron activity may not be present for an individual with autism, resulting in them not having the capability to understand and calculate the why behind many aspects of behavior. Mirror neurons are found in the inferior frontal gyrus and inferior partial lobe. The human mirror neuron encompasses the posterior partial cortex and the ventral premotor region. Grays et al. Recorded when recording emotional gestures no substantial activation of the amygdala or the inferior frontal gyrus in participants with autism, whereas control participants depicted significant activation here. Transcranial direct current stimulation can be defined as the neuromethodology non-invasive brain stimulation tool that includes a low directed current of electricity to mutate neurons in the human brain. This technique alters brain physiology as well as psychological, motor, and behavioral processes. This is achieved by adjusting cortical excitability. Unlike other non-invasive brain stimulation techniques such as transcranial magnetic stimulation, tDCS does not completely generate cerebral activity. Instead, it modifies instinctive brain movement and excitability. This is achieved by sub-threshold modulation of neuron membranes, furthermore stimulating long-lasting variations of cortical excitability and activity. This technique results to be significant as these neuroplastic developments are vital fundamentals for severe cognitive functions, such as learning, memory formation, and also pathological transformed in frequent neurological and psychiatric diseases. In this 2018 study, the researchers recruited 13 subjects, 11 being males and 2 females. All of these subjects accepted 10 times tDCS modulation over the course of 3 weeks. The researchers also recruited 13 subjects who remained for the experiment as control groups. All subjects that participated in the study were diagnosed to be ASD by a professional psychiatrist. During the procedure, the anodal electrode was positioned over the durastral prefrontal cortex and the cathode electrode was placed over the right supraorbital, allowing a direct current of 1 mA to be used in a battery-driven constant stimulator. This research predicts that repetitive restrictive behavior is likely to be generated by repetitive restrictive neural network dynamics in children with autism, which consume minimal entropy than typical developmental in children. Findings in this present study suggest that tDCS do in fact modulate brain motion during an adult tDCS intervention over the durastral prefrontal cortex has the potential to enhance the cortical excitability for individuals with ASD by boosting the excitation and reserve of neurons. This method depicts the findings that has tDCS can further enhance areas of cognition that regard to learning acquisition and performance to learning acquisition, and furthermore indicating that tDCS can be an effective and valuable tool for rehabilitation of individuals with ASD. Complementing the above study, in this 2018 study, the researchers wish to depict that transcranial direct stimulation is a processing, non-invasive method in autism. A systematic review was conducted. Electronic and manual searches were performed to detect studies published in peer-reviewed scientific journals tackling the use of tDCS in ASD population. This review included 19 studies. 11 studies were directed on children with ASD, while 7 were focused on adult participants. Results found that 5 studies contained neuropsychological functions outcomes focusing on language, executive functions, memory, attention, and motor skills. These findings concluded that tDCS is a hopeful method for the decline of ASD symptoms as well as for improving neuropsychological functions. This systematic review also depicted the findings in regard to clinical impairment. tDCS indicates substantial enhancement in socialization, repetitive behaviors, sensory and cognitive awareness. As stated in the above 2018 studies, further research is necessary to demonstrate the optimal treatment for tDCS for individuals with autism. Further research should focus on examining aspects to interpret the mechanisms of these treatments using neuroimaging techniques. Further research should also be focused on conducting greater varied control groups as the control condition varies across studies, with the sham condition being the most utilized. Finally, the undesirable side effects of tDCS should be reported in the future research to depict calculated quantitative derived measures more easily. It can be concluded after analyzing and discussing literature on autism spectrum disorder, the treatment of tDCS is a valuable method of treatment, in particular enhancing socialization and cognitive processes.